I'm a virologist, so we start with a picture of a bit of a virus at least. And I just want to give you a little bit of background before I go on to the bit about variants. So COVID or SARS-CoV-2 is an envelope virus. Its replication is and a lot of the intervention is on that initial step of binding and entry into the cell. And that's a two-step process for the virus. The first is that its receptor binding has to bind to the ARC2 receptor on the cell surface in the organs that express that. And then you have to have cleavage of the protein in order for fusion to occur and for entry into the cell. In the primary target tissues, like the respiratory tract, there is the presence of a protease called TMPRSS2, which aids the cleavage of the binding protein and facilitates entry. In other tissues, there are other proteases which will do that, but not as efficiently, which is an important determinant of what the primary target sites are. So a lot of what we talk about in variants relates to that receptor binding domain and other bits of the virus around that protein. And that's a picture of some of the areas earlier in the pandemic where these mutations were occurring around there. That yellow bit there is the receptor binding domain, and certainly most of the important mutations occur there or very near to it, and particularly that N-terminal domain, which is the red bit next to it. The other area where you can see mutations of biological significance are down there around that S1, S2 cleavage site, which is the step that's required for fusion. So we have a lot of different names for the mutation. On the right-hand side there, you can see the various colors that relate to different bits of the spike protein and the numbers of the residues that we use in naming the mutations. Okay. Now, in fact, SARS-CoV-2 has quite a stable genome, and we found quite early on that if you wanted to distinguish one strain from another, you weren't going to get it by limited sequencing. There wasn't enough variation occurring. So as you all know, there was a big explosion of whole genome sequencing as a result of the pandemic, because we needed to look at the whole 30,000 bases in order to get a sufficient distinction of different strains. Early on in the pandemic, there wasn't a lot happening genomically at all, and in fact, the only significant mutation occurred was this D614G mutation, which occurred in the spike. It actually didn't occur at the receptor binding domain, but it was nearby, and it had a steric effect on to actually increase the affinity of binding and improve uptake into cells. Then later in 2020, there was a lot more variation occurring in the virus, and we began to see a much greater range of variants occurring. And that's when WHO introduced this concept of variants of concern, variants of interest, and variants under monitoring, which we'll talk about a little bit more. Also, the numbering system became incredibly complex, where we had long strings of numbers to indicate different strains. And so those were then grouped more into what were more biologically significant variants, and they were given then letters of the Greek alphabet, so we could talk about the alphas, the betas, the gammas, the deltas, and now up to the omicrons, rather than having to continue to give a long string of numbers. And that variants could then be grouped into these major clades or lineages or whatever people chose to call them. The other thing that happened during the course of all this is we saw a shift in the way variants were generated. Early on, it was based upon nucleotide changes that generated a change in an amino acid. And that was allowed us to say, well, that's different. And those were the so-called drift variants. What's happened later, and I'll talk again about that, is we're seeing recombinations and much bigger changes in the numbers of changes, and that's the so-called shift variants. The mutation rate, that's dangerous, I haven't got a timer here. This is going to be a guessing game. The mutation rate here, why do we get so many variants? Well, we get so many variants because we look for them very studiously in this virus. And it relates to a number of genetic changes per replication cycle. That all of the RNA viruses undergo more changes than we see in the DNA viruses, which tend to be quite stable. But in fact, SARS-CoV-2 is one of the most, and all the coronaviruses are amongst the most stable of the RNA viruses. The reason RNA viruses tend to vary more is they lack a proofreading function, which goes along and corrects most of the errors. The coronaviruses do have that proofreading function, which is why we didn't see the same degree of variation, why we needed whole genomes. But variants do occur. There are still some replication errors in that. The host cell responses actually do some editing of the virus as well. And there's a very high rate of what we call non-synonymous mutations. That means a mutation which actually produces a change in amino acid. So unlike many other viruses where a lot of them are synonymous, that is, they don't change the amino acid, so they don't really matter, it happens much more commonly with SARS-CoV-2. It also has a high substitution rate. That means overall, there is a very high rate of accumulation of mutations. And there were about 2 per month in the pre-recombination era. Now that recombinations are occurring, there are much bigger changes happening. And this led to this a whole stack of different classification systems. And these are one of the earlier ones that came from Gilead. When there were about 150,000 full genomes in their database, there are now millions in the database. And it allowed at least some sort of understanding of the genetics diversity of the virus. But there was a WHO classification system. Underneath, I've put the lines for the Pangolin system, which was another classification. Public Health England had their own classification of them. The next strain had a different classification system. So it all got a little confusing for people. And I don't know at the end of this you'll be free of confusion, but you might be marginally less. But this is the sort of thing that we began to build up. And this is a next strain, which is a very common platform for presenting what's happening genomically with the virus. And if you look at the colored squares over there, you can see both the numbers that the next strain classification system is, but they're kindly in brackets. They actually label it according to the Greek names and some of the other variant names from the Pangolin system as well. Oh, wait a second, that shouldn't have happened. And here, and what this allows you to do is build up these international maps of where the particular variants are occurring internationally in the different distributions. This mob here is all Omicron. And you can see what's happened here with the evolution of these lineages. These are older ones going through the alpha, beta, delta, etc. lineages. And we'll see that a bit more. So that begins to build up a pattern of what we see, which is a mixture of these sort of changes, which are major changes, and then gradual evolution of the viruses as within those particular major variants. And this is a different way of looking at it, the same data, but here you can see this was the original stuff before we began seeing more of these variants occurring. And then the Greek names started in the later 20s. These are the alpha, that's delta. There was beta was in there somewhere, but there was not very much of it. Those were the delta variants, and then now we're moving into the current phase, and this is all Omicron with two different forms. In the early stages, we largely saw these drift variants where there were a limited number of amino acid changes within the spike protein occurring, to one which is dominated by recombinants, where you get a bit of the genetic material from two different viruses when they co-infect a host, and therefore you can get transfer and creation of a large number of mutations in the spike proteins simultaneously, because you get the mutations from the one and the mutations from the other all together. And that's produced a significant change in how we look at the evolution of this virus and its significance. This is the WHO variants. So I mentioned that there were these three classes, and I put them slightly in the wrong order, but that's sorry, my pointer's given up the ghost. There we go. There we go. That's right. We have a group which are the variants under monitoring. These are lineages where there are changes in the genome and in the amino acid sequences which give you some concerns, usually because there's sort of mutation that might escape from control, but there's nothing to really get you worried about at the moment. The variants of interest over here are the ones that have possible evidence for epidemiological, pathological, or immunological features of concern. And then the variants of concern are the ones where there is a detrimental change in disease severity, immune escape, or significant changes in epidemiology that might make public health interventions less effective. And if we look at where we are at the moment with the three of these, this is a sort of cut down version of the genomic tree that I showed you before. And as we go through the evolution of this virus, they're variously labeled as viruses of viruses under monitoring, and sorry about the formatting changing there, viruses of interest and viruses of concern. And along here, you'll somewhere in there, you'll see Delta is in there. There were a few other ones, Theta and Iota, that were in there that never really went anywhere. And then right up here is the beginning of the Omicron. And you can see Omicron has become the strain now of virus. And then various of those have been labeled as variants of concern, variants of interest. Don't pay too much attention to that in any particular graph, because those classifications change depending on how they assess the characteristics. Some of these have disappeared and are not really present any longer, so they've disappeared from the list as well. And it's all based on this, and if you quickly scan that, you'll see where all the mutations are. Here's the current list of variants of concern and variants of interest. And and they're all Omicrons, okay, because that's all we're seeing at the moment. Of particular interest, which we'll be talking about, as it says, we did have a number of them that were listed as variants of concern, the BA.2s, the BA.4s, the BA.5s, all Omicrons. But these are those parental lineages aren't circulating anymore, so they go off the list. As you may have seen, this one comes actually as a table from the ECDC website. And there are actually currently no SARS-CoV-2 variants which meet the variants of concerns criteria. Now, having said that, some countries have their own classification of them, and they may still count some of these as variants of concern, and some of them may go back on later, depending on what happens. and that's the ones on the list. You can see these are all recombinants, and we'll hear more about XBB 1.5 and related ones, which include names you may have heard, like the EG.5, the XBBs, and this FE.1. And they're all related recombinant viruses. And that's all I'll say about that at the moment. Pardon? Oh, no. I don't count the time it took me to get to the podium. Okay, so what do we do with these? It's like a scorecard. Terrible. Okay, using these, so what are we made of variants? This was some of the early stuff from Australia when we were looking at tracking the entry. And whole genome sequencing was very useful in terms of tracking entry and movement. And what we saw down the bottom were our very early clusters of cases, and they were all associated with somebody who recently returned from an epidemic area, and we managed to squelch all of those. And what we could see up the top when we began getting a lot more local cases with the same variants was when we were getting escapes from the quarantine hotel and community spread. And that really was the beginning of the first wave for the east coast of Australia at least. And that was very useful in its information. This was another one where we were able to show the use of genomics and variant analysis to show transmission from two cruise ships, which are docked, the Ruby Princess and the Ovation of the Seas. It's all in the public arena. And then subsequently, when everybody went home to their states, including Western Australia, we could show the amount of transmission that occurred associated with the travel on the plane of both of those variants, particularly the Ruby Princess one. So very useful in understanding and showing that we were able to control the spread of that. It didn't go much further than that plane flight. So why would the virus want to have these variants? Well, there are a number of advantages to it. They relate particularly to being able to improve cell entry by either better binding or enhanced fusion protein, a better cleavage of the spike protein, delivering the virus to the places where it's more likely to be shed and transmitted, particularly the upper respiratory tract, increasing the stability of the virus when it gets into the community, and then the immune escape that may occur with changes. So immune escape has been one of the ones that we've been particularly concerned about, of course, because of loss of protection from natural infection or from vaccination, and also because it's a stepwise thing, increasing antigenic change, and of course the immune-based therapy and escaping from those, but also increases in severity of illness, hospitalization of deaths, resistance to treatments, and increasing the transmissibility. The indicators of immune escape particularly are normally predicted based on mutations in that S1 protein, particularly around the receptor binding site, and that would predict it may escape. But it doesn't always happen. As you know, there's been many mutations there that have not resulted in any significant change of the virus behavior. You can look at failure of vaccine-induced antibodies to neutralize the variants in the lab using either pseudoviruses or wild-type viruses if you have containment for those. And of course, increasingly, we're looking at what the cross-protection may be based on the T-cell responses as well. And I guess loss of vaccine efficacy or effectiveness in trials or in vaccinated populations, but it doesn't usually particularly get to that stage before we jump on it, hopefully. What's going on now? Omicron's a dominant variant, circulating globally since 2022. Actually, I can see it here, and fortunately, it's been associated with lower rates of more severe illness other than in those higher risk populations. We've moved, the original COVID-19 vaccines were monovalent, and you've heard a bit about those, and then we moved to combinations of the original ancestral strains with a BA1 or 4 or 5 in there. What's happened now is because of the increase of the recombinants, we're seeing a different group of viruses, which were touched on in the earlier talk, that are more distant from the ancestral strains. And these are this XBB 1.5 and the BA 2.86 and a couple of others, which are all sitting in that group. The importance of that one is that this phenomenon of and the vaccine design and why they're going to a monovalent vaccine for those is that there's this concept of original antigenic sin, which is that when you give a vaccine that you want to deliver for these new variants, if people have had previous vaccination or natural infection, it actually stimulates preferentially the response to those older strains, and then not very cross-reactive to the newer ones, which is why we're looking now at monovalent vaccines when you think, well, why don't you just put them all in? Because it's counterproductive. So that's what we're moving to there, moving now into that next generation of vaccines, and those sort of changes I think are going to happen more often. Variants in pathogenicity. Generally, as I said, we've had a reduction of pathogenicity. This can be a long minute. A lot of variants of pathogenicity. It's tended to be attenuated, and Omicron is definitely less severe than the previous significant variants. But that's no guarantee that will happen. There's sort of this assumption that the virus will mutate towards being less severe because it doesn't want to kill its host. Well, that doesn't work if it kills its host after it's transmitted. So we don't want to be overconfident that that there's a pressure for it not to be more severe. And these, I will go through these very briefly. But the other area and significance of the variants is in the use of therapeutic monoclonal antibodies. And really, the ones we have currently, so Trivimab, certainly we don't use it in Australia anymore. And the combination one, which I keep forgetting the name of, is also losing effectiveness. And now there's certainly for the US and for Australia, we're really not using them very much at all, unless and it's only for areas where you don't have those resistance mutations. And you can't read them, but there are an awful lot of resistant mutations, and the Omicrons largely have got those mutations present in them. Doesn't mean newer ones won't come along or combinations, but it's something that's we're seeing. And there are some variants which are showing, there are variants that have some resistance to the antivirals, remdesivir, molnupiravir, and Paxlovid. And that's likely to become more problematic, but at the moment they're really not appearing at much of a significant rate in terms of therapeutic or prophylactic use. So I think Paxlovid's bit is the workhorse for us, certainly. And so what next? And this is the last slide. We expect there to be a continuation of drift and shift mutants to occur. It's the nature of the beast, and we will continue to drive those. We hope those variants will cause milder illness, but there's certainly no guarantee, and there's no absolute contraindication to illness becoming more severe. So we may get significant escape from vaccine, natural infection, or vaccine-induced protection with those effects. Resistance developing to antiviral drugs, the more we use them, of course, the more risk is of that of emerging them. Loss of access to vaccines because we don't have any that work anymore. And increased severity may still occur. And of course, we've got to watch for the emergence of new zoonotic coronaviruses with pandemic potential. So lots for us to continue doing. So thank you.
David. A question online. You indicated that some of the mutations give an idea of immune escape. Are there any mutations that you think are associated with more severe disease? Is that sort of the virologist can tell us, yeah, this mutation is likely, or do we wait until everyone's dying from it and then say, well, that one's severe?
In some ways, some of them will, the ones that enhance virus uptake will increase disease because you'll get a bigger viral load. So the 614G, But the beta, the alpha, the beta, the deltas were all associated with increase of severity of disease. So for some of them, it was a tendency to go to more, the migration down into lower respiratory tract from upper respiratory tract was greater. I don't know that we can describe the mutations that occur in there and some of the differences. With none of the mutations can you in itself say that they are going to do anything in terms of escape, because we've seen so many mutations that you think, oh, well, that might be immune escape, and you don't see any realistic immune escape. So I think there's a little way to go. Others may know, and I suspect it's going to come to that, how we learn to put together combinations that are going to be more predictive. But in the end, knowing whether it increases disease severity is based on finding out whether people get sick more often.
Yeah, so still a clinical question on the floor.
To what extent, right over here, to what extent can we predict the mutations? Obviously not completely, but is there any extent to which we can? I mean, the example I think of is the F456L mutation seemed to take everybody by surprise earlier this year.
Yes, look, I can't answer. All I can say is you think we should be able to, except to me, and this is getting out of my expertise, but to me it seems we describe a lot of mutations that put it into that, they go into the viruses of interest or the viruses under monitoring, because something's happening that we think may make it worse, but we don't actually classify them until something happens. We're still waiting for the epi. We're still waiting for that real life events to occur. I guess we're going to build up that knowledge base, and there'll be clever people who'll put them all together into some sort of model, I'm sure, eventually. But it's a wait and see at the moment.
Thank you. Thanks, Raja. Thank you, David. Oh, sorry, Raja, we've got one more. Can we quickly have a question, or do you want to? Yeah, sorry, just if we can quickly, David.
Just a quick question. Just want to know your opinion about regarding the natural characteristic of the virus that is mutating. What might be the best recommendation for getting the vaccine? Is it like influenza vaccine that you have to change, or you have combination of different strains? What do you think? Thank you.
Yeah, look, I think one of the reassuring things, I mean, the big change at the moment is going to a monovalent one because of the way the virus is behaving. And it's got a lot of parallels with the flu vaccine, which is that if we give a vaccine which is mainly hemagglutinin based, then you get this build up this huge memory cells to the hemagglutinin, and not to some of those stem loop ones which may give you broader protection. And what we see then is maybe you're not generating the antibodies to the right bit. So part of it is looking at how your vaccine, the type of response your vaccine is going to induce based on what you know or you think you know about the immune memory people may have laid down. So I don't know, you can say in the future exactly what's going to do it, but you've got to test them on that basis. Cross-neutralization is important, but we know that the T-cell epitopes are more common across the variants. And others may know, but I don't think we've really integrated that well into our understanding of what we would want from a vaccine, but we will. So currently, I mean, even if you look at these very new variants, if you've had 2 vaccines and a natural infection, or 3 vaccines, then we still have reasonable protection, probably even against these new variants. It's just not as good as we'd like. When you go to your XBB 1.5, which is now been authorized in a couple of countries, you're going to get better protection against those variants. But you're not, it's a sort of stepwise game. It's not a completely different vaccine. And it is very like we're seeing with flu. So they are going to have to retune the vaccine. whether it's going to be, and we assume it'll be an annual vaccine, but it might have to be a 6-monthly vaccine.
Thank you. Thanks, David.